Good day everyone. Before we dive into our topic, let's first understand what GMO is. A genetically modified organism or GMO is any organism whose DNA has been intentionally altered using genetic engineering. This means scientists can transfer, edit, or insert genes to give an organism new traits, something that wouldn't happen through natural mating or recombination. For example, a plant can be modified to resist pests or to produce more nutrients. So now, why are GMOs important? They play a big role in solving real-world problems in food, health, energy, and the environment. First, for food security. GM crops can grow faster, survive droughts, can resist pests, and help ensure a steady food supply. Second, for health. Through a process called biofortification. Some GMOs are enriched with vitamins and minerals to fight malnutrition. A great example is golden rice, which contains vitamin A. Third, for energy. GM plants or microorganisms can produce renewable fuels called biofuels, reducing our dependence on fossil fuels. And lastly, for the environment. Certain genetically modified microbes can help clean up pollution in a process called bioremediation, helping restore ecosystems. So GMOs comes in three main types: plants, animals, and microorganisms. First, we have genetically modified plants, which are the first common. This includes crops like soybean, maize, cotton, and canola, which are made to resist pests, tolerate herbicides, or have improved nutrition. Second, genetically modified animals like Aqua Advantage salmon, which shows faster than normal salmon because a transferred growth hormone gene. And third, genetically modified organisms such as bacteria or yeast, which are used in medicine, biofuel production, and environmental cleanup. For example, a modified E. coli produces human insulin used by diabetic patients. So the journey of GMOs started in 1973 when scientists Herbert Boyer and Stanley Cohen created the first genetically modified organism by inserting DNA from one bacterium into another. Then in 1982, the first GMO product, which is the human insulin made through recombinant DNA, was approved for medical use. And by 1996, GM crops were commercialized, starting with only 1.7 million hectares planted. Over the years, that number has grown tremendously, reaching around 190 million hectares across 24 countries by 2017. So today, major GM crops include soybean, maize, cotton, and canola, and many of these are grown by small farmers in the developing countries. Now, let's move to biofortification. Biofortification is the process of improving the nutritional quality of food crops using genetic modification, conventional breeding, or agronomic methods. Its main goal is to increase important nutrients like vitamin A, iron, and zinc in staple crops such as rice, maize, and wheat. This approach helps fight what we call the hidden hunger, a type of malnutrition where people get enough calories but lack vital vitamins and minerals. So there are two main ways to achieve biofortification. First is the conventional breeding, which means crossbreeding naturally nutrient-rich varieties with other high-yielding crops to produce new varieties with better nutrition. Second is the genetic engineering, or the transgenic approach, where scientists directly insert genes responsible for nutrient production into the plant's DNA. This is faster and more precise especially when the nutrient doesn't naturally exist in the crop. So, the nutrients most commonly targeted through biofortifications are first, vitamin A, which prevents blindness and strengthens the immune system. For example, golden rice and vitamin A-rich sweet potato. Second is the iron, needed for healthy blood. Deficiency causes anemia. For example, iron biofortified beans and pearl millet. Third is the zinc, which supports growth and immunity. Example, zinc-enriched rice and wheat. Fourth is the folate and protein quality, which help with fetal development and overall nutrition. So these nutrients are chosen because their deficiencies are widespread, especially in developing countries. One of the best examples of biofortification through genetic engineering is golden rice. It was developed in the 1990s by scientists Ingo Potricus and Peter Bayer. Golden rice was engineered to produce beta-carotene, which our body converts into vitamin A. Normally, rice grains don't have these nutrients. So to make this possible, two genes were added: the phytonin synthase from maize and the carotene desaturase from a soil bacterium called Erwina uredovora. These genes activate the carotenoid biosynthetic pathway in the rice grain, giving it a yellowish color, hence the name golden rice. So golden rice was approved for food, feed, and processing use in the Philippines in 2021, making our country the first to authorize the commercial planting. It has also been approved in Australia, New Zealand, and Canada. However, despite its scientific success, golden rice has faced controversies. Some critics argue that promoting dietary diversity and supplements might be better solution, while supporters believe it's a humanitarian innovation that can help save lives, especially among children suffering from vitamin A deficiency. So in genetic biofortification, the process generally involves several ways. First is the gene identification, finding the genes responsible for nutrient production. Second is the gene insertion, using tools like agrobacterium or gene editing to insert these genes into the crop's DNA. Third is the expression targeting, making sure the genes work in the edible part of the plant, such as the rice and the sperm. Fourth is the metabolic optimization, adjusting pathways so nutrients are produced efficiently without harming yield. So biofortification offers several key benefits. It provides a sustainable way to deliver nutrients through everyday food. It improves public health, reducing cases of blindness, anemia, and growth stunting. It is also cost-effective since it only requires planting improved seeds. It's localized, targeting the nutritional needs of specific populations. And lastly, it is safe and scientifically regulated, ensuring it meets food safety standards before release. And of course, there are also challenges. These are regulatory delays, which it takes years for testing before a GM crop is improved. There's also public skepticism. Many people still fear GMOs due to misinformation. Intellectual property rights can also make technology less accessible to poor farmers. And finally, there are ecological and socio-economic concerns like possible gene flow or uneven benefit distribution. So while biofortification is promising, it still requires proper education, safety checks, and policy support to be fully accepted. How does biofortification helps public health? So okay everyone, now let's talk about biofuels. So what exactly is biofuel? Simply put, biofuels are renewable energy sources made from living things. That means plants, algae, and even microorganisms. Instead of relying on fossil fuels like coal or petroleum, biofuel give us energy from materials that can grow or reproduce naturally. So there are main types of biofuels you might hear about. First is the bioethanol, usually made from sugars and starches in plants. Biodiesel, made from plant oils or animal fats. And biogas, which produced by breaking down organic matter without oxygen. And also drop-in fuels, which engineered fuels designed to directly replace gasoline or diesel. The goal of all these biofuels is to reduce greenhouse gas emissions and lessen our dependence on fossil fuels, which are harmful to the environment and limited in supplies. So now, let's talk about how we actually produce biofuels. So there are several production routes. First is the fermentation of sugars and starches. So basically, sugars from plants like corn are converted into ethanol by microorganisms. Second is the transesterification. It is a chemical process that turns plants' oils into biodiesel. Third is the anaerobic digestion, which microbes break down organic matter without oxygen to produce biogas. Fourth is the enzymatic hydrolysis, which are enzymes used to break down complex biomass into simple sugars or fermentation. And lastly is the oil extraction from engineered crops or microalgae. So some crops or algae are specially developed to produce more oils for biofuels. How does genetic modification help improve biofuel production? Biofuels are also grouped by generation, which basically tell us what raw materials they use and how sustainable they are. First generation. These are easy to process but compete with food. Example, corn, sugarcane, soybean. So if we use too much fuel, people might not have enough food to eat. Second generation, more sustainable because they use non-food sources like crop residues, wood chips, or grasses. These are harder to process but better for the environment. Third generation, these are futuristic, fast-growing, high oil yield organisms like microalgae and cyanobacteria that can grow on land unsuitable for crops. They don't compete with food at all and can produce a lot of fuel quickly. Science really helps make fuels better and more efficient. So there are some main technologies used. First is microbial fermentation, which microbes convert plant sugars into ethanol or butanol. Second is the enzymatic hydrolysis, which enzyme breaks down cellulose from plants into sugar that microbes can ferment. Third is the transgenic oilseed crops. These are genetically modified to produce high-energy oils for biodiesel. Engineered algae, yeast, or bacteria. This can also be modified to produce more lipids or fuel precursors. So how does genetic modification help? It boosts crop yield and makes plants or microbes more resistant to stress. It optimizes metabolic pathways so ethanol or biodiesel production is faster and more efficient. Engineered microbes can secrete enzymes that make biomass breakdown simpler. Tools like CRISPR allow precise gene editing to improve fuel production even further. Basically, GM crops technology makes biofuel production faster, cheaper, and more sustainable. So why should we care about biofuel? So well, there are several benefits. First is they have high oil yield, meaning we can produce more fuel from less raw materials. Second, they don't compete with food production, especially in second and third generation biofuels. They help reduce greenhouse gas emission, which is used for fighting climate change. Biofuel production can produce rural development, creating jobs in farming, processing, and technology. So it is not just good for the environment, it can also benefit communities. And of course, biofuels are not perfect. Some challenges include high production cost, making biofuel is still expensive compared to regular fuels. Biosafety and containment of GMOs. We have to make sure genetically modified microbes or crops do not accidentally affect the environment. Next is the limited biomass supply. There is only so much raw material available. Some first-generation fuels have weak greenhouse gas benefits, meaning they are not always better than fossil fuels. Lastly is the regulatory and public acceptance issues, because some people are sometimes skeptical about GMOs or new fuels. Now, let's talk about how GMOs can help clean up for our environment. Bioremediation is basically using living organisms, usually microorganisms, which is to remove or neutralize pollutants from soil, water, or air. Think of it as a nature cleaning crew, but enriched with science. So there are two main ways it is applied. On-site, which is cleaning pollution right where it is, while and off-site, which is moving contaminated materials somewhere else to clean it. It is a safer and more eco-friendly alternative to chemical cleanup, which can sometimes create more problems than it solves. Closely related is the Phytoremediation, which specifically uses plants instead of microbes to clean the environment. Plants can absorb pollutants through their roots, sequester them in tissues, volatiles, harmful compounds, or transform pollutants into less toxic forms. For example, some plants can take up heavy metals like lead or cadmium from soil. This is mostly done on-site, meaning the plants clean the soil without needing to move it anywhere else. What is the key difference of bioremediation and phytoremediation? Here's the genetic modification comes into play. Scientists can engineer microbes or plants to be even better at cleaning pollutions. Some ways GM helps. It makes microbes better at breaking down toxic substances like oil spills, pesticides, or heavy metals. Speeds up environmental cleanup because it is faster and safer than traditional chemical methods. Tools like CRISPR or gene cloning to enhance genes responsible for pollutant removal. It creates synthetic microbial communities that work together to clean up pollutants more efficiently. It also helps organisms survive in harsh or polluted environments, so cleanup can happen in tough conditions. Basically, GM technology makes environmental cleanup more efficient, cost-effective, and sustainable. Let's look at some real examples to make this more concrete. First is the transgenic plants like Nicotiana tabacum or tobacco. Next is the Arabidopsis thaliana and the Beta vulgaris were engineered with bacterial mercury reductase genes called the MER-A or MER-B to detoxify methyl mercury in polluted soils. Microbial consortia, groups of microbes like Enterobacter cloacae, Sporoxarciana soli, and Viridibacillus arenosi, can work together to remove heavy metals such as cadmium, lead, and copper. These combinations are much more effective than single-strain treatments. So instead of one organism doing all the work, we have a team approach powered by science. These methods have several benefits. Low cost because it is cheaper than chemical cleanup or heavy machinery. On-site remediation because no need to move contaminated soil or water. It is also less disturbance, natural process so ecosystems are not disrupted. Potential for recovery because metals or other substances can sometimes be recovered. Healthier ecosystems, which reduces risk to humans and wildlife. Of course, there are challenges to keep in mind. First is the containment and ecological risk, which releasing GM crops, microbes, or plants into the wild could affect other species. GM flow, which engineered genes might transfer to other organisms. Long-term monitoring, which we need to track sites to make sure cleanup is safe. Regulatory acceptance, which government approval can take time, and public acceptance is sometimes tricky. When scientists do these projects, they need to carefully measure and monitor outcomes. Removal efficiency, which how much pollutant was actually cleaned. Time to remediation, on how long did it take. Biomass disposal plan, on what happens to the plants or microbes after cleanup. Containment and ecological risk assessment, making sure nothing harmful spreads. Field trial outcomes, comparing effectiveness under real conditions. They also use molecular methods like PCR or DNA sequencing to confirm that the engineered genes are present and functioning properly. This ensures safety and efficiency before real-world application. Alright, now let's talk about how scientists actually test and work with GMOs before they can be used. First, there are molecular methods. Scientists use tools like PCR, qPCR, and DNA sequencing to detect and confirm the presence of GMO genes. So why do we need this? Because it helps us identify the exact genes, constructs, and promoters used in genetic modification. It also ensures that the inserted genes are stable and working properly in the organism. For example, scientists might use GMO detection PCR primers to confirm a gene is present in a plant. This step is super important for verifying the genetic identity and for biosafety before the GMO is released into the field. What are molecular methods like PCR and DNA sequencing used for GMO development? Once the molecular checks are done, scientists move to field trials, which are carefully planned experiments to test GMOs in real conditions. Field trials are done under strict containment and biosafety protocols to avoid any accidental escape to GMOs into the environment. Usually, they use something called Randomized Complete Block Design or the RCBD. It is a fancy way to make sure the experiment is fair and reliable. Trials focus on both agronomic traits like plant growth or biomass and nutrient traits such as an iron or vitamin A in crops. Environmental monitoring is also included to make sure no harm is done to surrounding ecosystems. So, how do scientists measure success? It depends on the GMO project. For biofortification, they measure yield, plant growth, and nutrient content like iron, zinc, or vitamin A. And for biofuels, they check biomass amount, oil yield, and fuel conversion potentials. And for biofortification or phytoremediation, they evaluate pollutant reduction, soil recovery, and water quality. So in general, they also look at overall performance, stability, and environmental benefits to make sure that GMOs does what it's supposed to do without negative side effects. Science is not just about the lab, it is also about people and communities. So ethical research is the key. Before trials, scientists obtain consent from local authorities and communities. They conduct stakeholder consultations with farmers, scientists, and policymakers. Information campaigns are held to explain the project goals, safety, and benefits. So why it is important? Because it promotes transparency, trust, and ethical responsibility in GMO research. People need to understand what is happening and why it is safe. And of course, GMOs are highly regulated. Here's are the framework. First is the National Biosafety Law, which every country has law to test and approve GMO before they are used. They make sure GMOs are safe for health and the environment. Second is the Cartagena Protocol on Biosafety. It is an international agreement that ensures countries safely handle, move, and use GMOs across borders. Next is the Food Safety Assessments, which scientists check for toxicity and allergenicity from GMOs are consumed. Next is the approval process, which the GMO goes through many tests before they can be planted or sold. Even with regulations, there are social concerns. First is the farmer seed sovereignty, because some farmers worry they cannot save or reuse GM seeds because companies own the technology. They want control over their own seeds. Corporate intellectual property or IP. There are large companies owning GMO patents can make technology expensive for small farmers. Next is the public acceptance, which some people distrust GMOs, fearing harm to health or nature. Education and awareness are essential to address this. Next is the misinformation. There are many false stories claiming all GMOs are dangerous, which is not true when properly tested. Lastly is the benefit sharing. It is important that the benefit of GMOs are shared fairly, especially with farmers and countries that need them the most. And finally, some big questions policymakers have to consider. First question is, should biofortified GM crops be fast-tracked in areas with severe malnutrition? Should we quickly allow GM crops in places where many people lack essential nutrients? Or what safeguards should be in places or release engineered microbes? And last question is, how can we make sure modified microbes used to clean up pollution do not harm the environment if they escape? So these questions show that science, ethics, and policy must work together to make GMOs safe, beneficial, and socially responsible. Thank you for listening and watching.